Good morning, students. In last class, we have discussed that is what are chromosomes and how they are segregated in human beings, where they produce the male and a female organisms. So that here, when their meiotic division is taken place, here only the half of the characters are transferring from parents to their young ones. So half of the character is from the mother and half of the characters are produced from the father, where here it producing a constant number of chromosomes remains a constant in an individual organism. So how here segregated, that means here males are heterogametic in condition, where here it shows that two different chromosomes are presenting, that is X and Y. And in females will having a homogametic, that means they are same type of chromosomes are presenting, but they are considered as one. So if the fuses with the X of the mother and X of the father, here female gametes are produced, and X of the mother and Y of the father, it is Y chromosomes of father. If they are fuses, here the male organisms are produced. Already we have discussed the males are the responsible for producing of either a male organism or a female an organism. Now coming next, now going into the next one, that is Mendelian and characters, that is a law of dominance. First one is the law of dominance. So here Mendel, there are three laws are presenting. One is law of dominance. Next one is a monohybrid, that is law of segregation. In this segregation, we'll having one is monohybrid cross, and second one is a dihybrid cross. So now coming to the first one here, that is a law of dominance. So here a law of dominance means here if out of the two characters, out of two homozygous characters, one of the character will be a dominant character and other will be the recessive character. So here will taken place a dominance, means out of the pair of contrasting characters, out of pair of contrasting characters, one is a dominant, one is a dominant, and another is a recessive. So here one will be the dominant character and another will be the recessive character. For example, if we take place an example, here an example, if we take place, there are 7 pairs of characters are presenting. In any of the 7 pairs of contrasting characters, we can take place any one. For example, if we take place the axial and the terminal, that means the position of the flower. If we take place example, the position of a flower. So if we take place a position of the flower, one is axial and second one is a terminal. Here axial will represent capital A, and terminal will represent the small a. So here the gametes are capital A, and here the gametes are, that is we are producing the gametes. Here small a will presenting. So when they are segregated, here it producing capital A, small a, where here we can call this as an F1 generation. F1 means we can write first filial generation. F1 means a first filial generation, where here it is an heterozygous condition. And heterozygous a zygous, it is a heterozygous conditions are taken, that is a heterozygous and axial plants, axial flowers are presenting. Why means here A is the dominant over the recessive. So here the recessive character will not express their character over the dominant, so that only a dominant character only will be expressed. So if you say in here F1 generation, here in F2 generation, if you see in F2 generation, if these are self-breeding is taken place, here the self-breeding. So when here taken place a self-breeding, here we can able to produce, that is capital A is crossed with capital A, small a is crossed with self-breeding. So that here what happens, these are the two gametes. So one is male and a female gametes. So here we have to know about the checkerboard. The checkerboard. What is the checkerboard means? It is the representation of here it was introduced by an R.C. Punnett. It was introduced by the scientist an R.C. Punnett, where he is a mathematician. A mathematician. He is a mathematician where he can introduce the checkerboard, where in the checkerboard one side will taken place as a male gamete. On the checkerboard, here one side will be taken place as this is a checkerboard. Here if we taken place one side we are taken place the male gametes, and another side we are taken place the female gametes. So when you are taken place here capital A, capital A, capital A, small a, capital A, small a, and small a, small a. So here we can able to produce an axial, here heterozygous, axial, here also heterozygous axial, and here it is the terminal. So here this shows that these are the dominant characters. That means here this law will shows that the law of a dominance. What is dominant means? Out of a pair of contrasting character, one is the dominant and another is the recessive. The dominant character will express over the recessive. So that your dominant character only will express not the recessive character. So this law we can call as a law of a dominance, where here an example we can take in place, that is here out of these three, here one is purely an axial condition and two are a heterozygous and axial heterozygous, that is heterozygous axial condition, and one will is purely a terminal. So here when you see in here a dominant character is expressing, that means three dominant characters are expressed and only one will be that is the recessive character will be presenting, where here phenotypic will represent 3:1 ratio and a genotypic ratio will be taken place 1:2:1 ratios. So here we cannot take the dominant, that means here we cannot take the recessive, that is genotypic as well as the phenotypic. Why means here we are taken place only a dominant character only, dominance, that is it represents the dominant character over the recessive, which was explained by the Mendel, which where you taken place any of the 7 contrasting character, that is axial terminal, a tall draft, round yellow or yellow green. Here these are the different pairs of a contrasting characters where they are taken place. The Mendel have taken 7 contrasting characters. Out of these, if you taken place any of these, here a dominant character will express the dominance only. Here the recessive character cannot express over the dominance, then we can call that as a recessive character. A dominant character means it is the character expressed over the one of the contrasting character and express dominant over the recessive, then such condition we can call that as a a dominant a character, or we can call as law of a dominance, which was explained by the Mendel. An example we taken place the axial and the terminal. Next, coming to the second one, that is, if you say a second one, that is here second one, if it taken place, law of a purity of gametes. Second one is the law of a purity of the second one. The law of purity of gametes. Or otherwise we can use the word as the monohybrid cross also. A monohybrid cross. So here, why we can call as a law of a purity of gametes or a monohybrid cross, we can call as a law of a purity of gametes, means here we can able to produce what are the characters we are taken place purely, that characters are produced in the young ones. So we can call that as a law of a purity of purely the gametes. What are the gametes we are segregated, the same gametes are produces, so that we can call that as a law of a purity of gametes, or otherwise we can call that as a monohybrid cross. So here, what is a monohybrid cross means, if a plant or animal, if a plant or animal, if it is a plant or animal segregated, if a plant or animal obtained by crossing over a homozygous parents or crossed over, here we are able to produce only one character is producing. If a plant or animal, if a plant or animal obtained by the crossing of homozygous parents, which differs in one character, then it is called as a monohybrid cross. So here in monohybrid cross, if you taken place an example if you taken place, here the two homozygous parents are, one is the tall character is crossed with the dwarfed character. So here these are the two homozygous parents. One is a tall character and another one is a dwarfed character. So here these are represents capital T, and this represents with the small t, where the gametes, here we are able to produce in capital T and we are able to produce small t. When these are segregated in F1 generation, here we are able to produce capital T, small t in F1 generation. a generation. So, what are the F1 generation it is produced? Here it is an heterozygous tall character. That means when two homozygous parents are crossed over, in F1 generation we are able to producing the heterozygous a tall character is produces. So when this heterozygous tall character is self-breeded, it is a self-breeded, then in F2 generation we are getting the phenotypic as well as genotypic ratios we are able to produce. So in it is a self-breeded, if it is a self-breeding, if it is a self-breeding in F2 generation, in F2 generation we are able to produce, that means capital T, small t is crossed with capital T, small t, where here these are the gametes where we are taken place checkerboard, capital T, small t, in the presence of a checkerboard, if you taken place a checkerboard, here capital T, small c, this is one side male and another side is a female. So here in checkerboard already said one side you have taken place male gametes and another side you have to take a female gametes. So here capital T, small t, capital T, small t as well as small t, this small t. So here purely it is homozygous, one is purely homozygous tall and this is heterozygous tall and it is also a heterozygous tall and purely the homozygous the recessive character, the homozygous a draft. So here it is homozygous, the tall character, homozygous tall character and heterozygous tall character and homozygous draft characters are produced, where here if you take in place that is a phenotypic ratio. So here we know that a phenotypic ratio means the external appearance of the a gene. It is an external appearance of a gene we can call as a phenotypic ratio. So here phenotypic means here three tall characters are produced and one recessive character. That means three tall character will express the phenotypically and one will be the recessive character. And if you take in place the genotypic ratio. So here genotypic ratio will be the 1:2:1 ratio. That means we will having one is purely a homozygous tall character and one is purely homozygous tall character and two are a heterozygous tall character, and one is purely a homozygous recessive character or draft character. So here purely one dominant character and two are heterozygous tall characters and one is purely a homozygous the recessive character it is produced, that is the draft characters are produced. And here already said the monohybrid cross we can call as a law of purity of gametes. Why we can call as purity of the gametes means what are the gametes we are taken place, that is a tall and a draft. What is the gametes we are taken place? So in the name only purity of gametes. That means purely the gametes. What are the gametes we are taken place at the end of this in F2 generation? In F2 generation, here purely we are able to producing the tall and a draft characters only we are able to producing. No other characters are not be produces, so that here monohybrid cross we can call as the law of purity of gametes. This is how explained, that is, it is called as law of purity of gametes. What are the gametes we are taken place in the gametes, the parental gametes, that is homozygous parents which are taken place, that is the tall and the draft at the end of the gametes. That means in F2 generation also. In F2 generation also we are able to producing the tall and the draft character only we are produces, not any other characters. So that here this law we can call as the law of purity of gametes. This is a second law which was explained by the Mendel, that is law of purity of gametes, or otherwise we can call as the monohybrid cross. That is, if you are, when two homozygous parents are crossed over, here we are able to get only one character we are getting, that is either tall or a draft character only we are getting. So that this is we can call as the monohybrid cross. Mono means a single, hybrid means when two parents are crossed over, here we are getting only one character only we are producing, so that either tall or a draft character only. So this is called as the monohybrid cross, or a law of a purity of gametes. This is the second one. Next, if you see the third one, that is the law of segregation, law of independent assortment. Next, the third one. The third one is the law of independent assortment. Or otherwise we can call this as the dihybrid cross. So here we can call this as the dihybrid cross. So here that is a law of independent assortment. Why you can call as law of independent assortment means when two homozygous parents are crossed over, here the independent, that means each character will express their own character. When they are segregated, each character will express their own character. Then we can call as an independent assortment. That means when they are segregated, when they are combined with one another in F1 generation, if they are segregated in F2 generation, we are able to produce each character will express their own characters. So that what happens, we are able to producing different characters are producing in an F2 generation. So that here we can call as an independent assortment, or we can call as a dihybrid cross. What is the dihybrid cross means? If a plant or an animal obtained by the crossing of two homozygous parents are crossed over, we are able to producing two pairs of contrasting characters are presenting. We are able to produce two pairs of contrasting characters are presenting, then we can call as a dihybrid cross. If a plant or animal obtained by the crossing of two homozygous parents, two homozygous parents produce two pairs of contrasting characters. So here when two homozygous, that means if a plant or animal are obtained by crossing of two homozygous parents, we are differing or produced, the parents differ in two pairs of contrasting characters. Produce or differs or produce two pairs of contrasting characters. So that is called as the dihybrid cross. So if you taken place an example, if you taken place that is a round and yellow, two pairs we are taken place, round and yellow is crossed with the wrinkled and green. So these are the two pairs of the two parents, the two homozygous parents are crossed over. So here one is round and yellow, one character, and second one is wrinkled and green, it is another character, where here dihybrid, two characters we are hybriding. So here when you take round and yellow is represented with capital R, capital Y is crossed with small r, small y. So here the gametes are the capital R, capital Y, and here the gametes are the small r and the small y. So in F1 generation we are produced capital R, capital Y, the small y, that is produced in F1 generation. So in F1 generation we are able to producing the heterozygous. Here we are able to produce the heterozygous round and yellow character is produced. That is in F1 generation we are able to producing an heterozygous, the round and yellow characters are produces. That is in F1 generation. So this F1 generation is a self-breeding is taken place. So here F1 generation, it is a self-breeding, capital R, capital Y, small y is crossed with capital R, capital Y, small y. So when these are forming, here one is female, one is male gametes, and another one is a female gamete. So that how you are crossing means capital R, capital Y, and capital R, small y, and the second one is small r, capital Y, and fourth one is the small r, small y. So these are the four gametes where they are produced of the male gametes. So here also we are able to producing capital R, capital Y, capital R, small y, and small r, capital Y, small r, capital Y, and lastly small r, small y. So these are the gametes where we are produced in the NF2 generation. That is gametes when you make a self breeding is taken place. So here we are able to produce these conditions. Now coming to this so coming to this Kepler, that means checkerboard. If it taken place the checkerboard, so if it taken place, one is Kepler's capital Y. Here one side male gamete and another side is a female gamete. So Kepler capital Y, Kepler small y. Next small r capital Y, small r small y. Here also same capital Y, Kepler small y, Kepler small r capital Y, small r small y. So when you are crossing here, we are able to produce the Kepler capital Y. Next Kepler capital Y small y, Kepler small r capital Y, Kepler small r capital Y small y. Next, if it is second one, Kepler capital Y small y, Kepler small y, Kepler small r capital Y small y. Next, small r Kepler small r small y. Next, if you see in Kepler small r capital Y, Kepler small r capital Y small y. Next, small r capital Y and small r capital Y small y. Next here Kepler small r capital Y small y, Kepler small r small y, small r capital Y small y, and purely here the small r small y. So here this is where we are able to producing the 16 gametes are produced. 16 gametes are producing when both here two homozygous parents are crossed over. In F1 generation we are able to getting the heterozygous round and yellow character is produced. When these are crossed over, we are able to getting the gametes 4 into 4. That means 4 male gametes and 4 female gametes are crossed over. We are able to get 16 gametes are produced. Out of the 16 gametes, here one is purely round and yellow. Here pure round and yellow, and last one is pure the wrinkled and green, and remaining 14 characters which are presenting when they are segregated. Here independently their own character is expressing. Here 14 are different recombinants. So here we can call them as recombinants, genetical recombinants we can use. Why means each character they express their own character will express independently during and segregation. So that here we can call as and dihybrid cross is also called as a law of an independent assortment. We can call that as the law of an independent assortment. We can call that as the law of independent. Why means each gamete will express their own character. Expect if two homozygous parents, if we remove remaining characters they are not present in the parents, each character will express their own character is expressing. So when they are segregated, so this law we can call as the law of independent assortment. So here if you see in the phenotypic ratio, the phenotypic ratio will be 9:3:3:1 ratio we are able to produce this. That is if you say that is round yellow, this is also round yellow, this is also round yellow, this is also a round yellow, this is also a round yellow, this is a round and a green, and it is also round yellow, here it is a round green, and here it is a round yellow, and it is a round green, here a wrinkled yellow, here also a wrinkled yellow, and it is a round yellow, and here it is a round a round green, a round yellow, and it is a round green, and here it is a wrinkled and the yellow. So that here we are producing 9, 1 round yellow, 1, 2, 3, 4, 5, 6, 7, 8 plus 9. So here we are able to producing 9 are round yellow, and 3 here round green 1, and here round green 1. So here 3 are the round green, and 3 are 1, 2, 3 are round wrinkled and green, 3 are the wrinkled and yellow, and 1 is purely the homozygous, that is a round and a green. That is wrinkled and a green. So here 9 are round yellow, 3 are round green, and 3 are wrinkled yellow, and 1 is purely wrinkled and a green are producing. So this is where we can call as the phenotypic ratio, that is externally appearance of the gametes are, that is 9:3:3:1 ratio, that is here taken place. If you taken place genotypic ratio, genotype, here a genotype will be the 1:2:2:1:2:2:4, 1:2:1 ratio. Here a genotypic ratio will be 1:2:2:4, 1:2:1 ratio, that is we can call as a genotype, where it is an internal structure of or internal constituent of a gene, that is internal structure of a gene is we can call as genotype, that is 1:2:2:4, 1:2:1 ratio, that is produced. How these are produced, we'll discuss now. One here purely 1 means purely the round and yellow, capital Y. So here there is no any other formation of the capital Y. So it is 1. And 2 capital Y, small y. This is 1. And if you see in another one, here it is 1. It is the second. So here 2 is 2 over. Next 2 capital small r, capital Y. So here it is 1. and here another one is 2. So here it is 1 and it is 2. So you can able to get 2. Next 4. 4 means capital R, small r. Sorry, capital R, small r, capital Y, small y. So this is 1 and this is 2 and this is 3 and this is 4. Capital R, small r, capital Y, small y. Here also the 4. Here we can able to produce that is 4 ratios. Next 1:2. 1:2 means capital R, small y. If you see in here, there is no any other one is produces. That is capital R, capital Y, small y. It is not produces. Next capital R, small y. So it is 1. Here 2. Here 1. It is 1. And second one is here capital R, small y. This is 2. So if it in here, 2 is also over. Next, if you see in here 1:2, that is small r, capital Y, small y. Sorry, capital small r, capital Y. Here there is no any other form. So here purely 1 only. So here 1 ratio. Next 2. Here 2 means small r, capital Y, small y. This is 1 and this is 2. So here we can also remove totally here small r, small y, capital O, small y. It is purely 1. So that here it shows that this is this phenotypic ratio of the a dihybrid cross, that is 1:2, 2:2, 2:4, 1:2, 1:2:1. This is where we can call as, that means here except the parental character, that is round and yellow and wrinkle and green, remaining 14 characters are produced independently. That means each character express their own character is expressing independently. They expressing when it is segregation is taken place. So that this law we can call as law of purity of gametes, or otherwise we can call as dihybrid cross. That means when you cross 2 homozygous parents are crossed over, we are differing in 2 different contrasting characters. So here differing 2 contrasting character, round and yellow, wrinkle, round and green, wrinkle yellow. So here 2 pairs of contrasting characters are differing, so that 2 different characters are differing, producing, then it is called as the dihybrid cross. So here 9:3:1 is the phenotypic ratio, and the genotypic ratio is 1:2, 2:4, 1:2, 1:2:1. How it is produced already we have discussed. This is of the 3 laws, that is law of dominance, law of purity of gametes or monohybrid cross, and lastly law of independent assortment or the dihybrid cross, and remaining we will discuss in the next class.